Today we are discuss about the topic of life history of Penicillium. So Penicillium, it is a genus of saprophytic fungi. They grow on dead decaying organic matters. So these Penicillium belongs to Ascomycetes. So these Penicillium members are economically used for the production of food materials and antibiotics and production of several enzymes. So these Penicillium play an important role as a decomposer in ecosystem. So the Penicillium grow on variety of habitats like soil, air, and few of the members are grow on dead decaying organic matters. Some of the Penicillium members are grow on rotten vegetables and rotten fruits, and they are grown on rotten meat. A few members of Penicillium used for the production of organic acids like citric acid and aspartic acid. These are organic acids for the production of these from the Penicillium species, and the few amount of species they are used as for the preparation of cheese. So this is called as a Swiss cheese. So these Swiss cheese produced from the Penicillium roqueforti, and this is commonly called as roquefort cheese. Penicillin, it is a antibiotic which is extracted from the species of Penicillium notatum. Now it is commonly called as a Penicillium chrysogenum. So Penicillium notatum are also called as a Penicillium chrysogenum. These penicillin, it is a wonder drug used in Second World War in 1929. So these penicillin extracted from the Penicillium notatum species, and these penicillin discovered from the scientist Alexander Fleming. Now classification of penicillin: kingdom fungi, division Ascomycota, class Uratiomycetes, order Urosales, and family Trichomaceae, genus Penicillium. In this Penicillium, a number of species are reported, in that a few members are given below. They are Penicillium notatum and Penicillium chrysogenum and Penicillium camemberti and Penicillium claviform, Penicillium aculeatum and Penicillium glaucum. These are the a few members of useful species in Penicillium. Vegetative morphology of Penicillium. So this is vegetative mycelium of Penicillium. In this mycelium, substratal portion which is called as vegetative hypha, and second one aerial hypha, that is called as conidiophore, and medullae, sterigmata and conidium. These are main parts in vegetative mycelium. In that, so first we are discussed vegetative hypha and aerial hypha. Vegetative hypha, which is creeps on the substratum, and it is uninucleated and septate wall, and branches are run in all direction on the substratum. So these substratal hypha secretes several enzymes. With the help of these enzymes, absorbs water and food materials from the substratum. And the second one, aerial hypha. Aerial hypha are cylindrical, stout in structure, and it is also septate and uninucleate. Conidiophores are developed on the aerial hypha. So, these conidiophores are multinucleated septate cells. Hyphal cells are uninucleated cells and the conidiophore cells are multinucleated septate cells. On the conidiophore, medulla which is present. Medulla is nothing but branched conidiophores are called as medulla. Now, the tip of the medulla, bottle-like structures which are present on the tip of the medulla, that is called as phylloids or sterigmata. So, these sterigmatal cells are uninucleated cells. Medullae and conidiophores are multinucleated, but the sterigmatal cells are uninucleated. Tip of the sterigmata, a number of conidias are formed in a chain like manner. Conidia is nothing but it is a spore. Non-flagellated spherical or oval structure spores are called as conidia. A number of conidia are formed in a chain like manner on the sterigmata. So, that is why penicillium is commonly called as painter's brush, because a number of conidia are formed and they are look like broom like manner. So, penicillium is commonly called as painter's brush. Now, structure of penicillial cell. So, this is penicillial cell structure. It is a eukaryotic cell structure. In this cell which is covered by thick cell wall, and these cell wall which is differentiated into three layered. One is the outer layer and middle layer and innermost layer. Outer layer which is composed of glucan materials and middle layer which is made up of proteins and a few amount of polysaccharides, and the innermost layer of the cell wall which is composed of chitin materials. Chitin, glucan and polysaccharides, these are main components of the cell wall. So, it is a thick cell wall, and this cell wall which gives definite shape to the cell. Below the cell wall, a living membranous structure which is present, that is called as plasma membrane. So, it is a living membranous membrane. Now, next, cytoplasm and protoplasm and all the cell organelles which are surrounds the plasma membrane. Cell organelles like nucleus, mitochondria, vacuole, endoplasmic reticulum, ribosomes, these are main cell organelles in the penicillium cell. In that, nucleus which plays an important role in all metabolic activities. So, this nucleus is uninucleate, it means only single nucleus which is present inside the cell, and the double membranous cell organelles are nucleus and mitochondria, both are present in this cell. So, chloroplast, that is absent, and ribosomes, 70S type of ribosomes are present, and these are scattered in the cytoplasm. Adjacent cells are, so these penicillium adjacent cells are connected with a simple pore, that is called septal pore. Adjacent cells are connected with the help of septal pore. So, this is cell structure of penicillium. Next, reproduction in penicillium. Penicillium reproduces by mainly three methods. One is the vegetative reproduction, and second one asexual reproduction, and third one sexual reproduction. Now first we are discussed what is vegetative reproduction. So Penicillium species vegetatively reproduces by fragmentation. Fragmentation is nothing but a portion of thallus which breaks several pieces, and each piece is known as fragment. So these fragments are germinate and produce new plant body, that is called as fragmentation. So by this process in Penicillium, parental plant body breaks several pieces and each piece is known as fragment. So these fragments further germinate and produce new mycelium. So this is one type of vegetative reproduction in Penicillium. Now the second one, asexual reproduction. So asexual reproduction with the help of conidia. So this is conidia structure. So these conidia are non-flagellated spores which are formed on the conidiophore. When we see the structure of conidia, conidia are oval, rounded in shape. It is covered by thick wall. In this thick wall, which is differentiated into two layers, one is the outer layer and second one inner layer. Outer layer is called as exine and the inner layer which is called as intine. In this conidium, outer layer which is further divided into three sublayers. One is the outer and second one middle layer and third one innermost layer. So this is exine which is differentiated into further three sublayers, outermost layer and middle layer and the innermost layer. So these all three sublayers which are made up of chitin, glucan and a few amount of polysaccharides. In this conidium, a single nucleus which is present, and that nucleus is a double membranous cell organelle in the conidia. Next, development of conidia. During the developmental stage of conidia, first sterigmatal cell which is a uninucleated cell. So these sterigmatal cell nucleus undergoes to mitotic division and forms two nucleus. Now the newly formed nucleus and few amount of protoplasm migrates towards the sterigmatal end. Now the septa which is formed between these two nucleus, now the conidia which is separated. By this process, a number of conidia are formed in a chain like manner. In each time, sterigmatal cell nucleus undergoes to mitotic division and forms two nucleus. So this is development of conidia. During the asexual reproduction in Penicillium, all the conidia are formed on the conidiophore. Now this, when the mature stage of Penicillium, conidia are fall on the suitable substratum, they germinate and produce new mycelium. So that is asexual reproduction with the help of conidia in Penicillium. When the asexual stage of Penicillium, after the liberation of conidia, they carried out by wind and they are fall on the suitable substratum. First of all, conidial wall elongates and produce a germ tube. So, by this germ tube, repeated divisions and forms a new mycelium. So, that is asexual reproduction in Penicillium. Now, next, sexual reproduction. In a favorable condition, Penicillium reproduces with the help of oogamous type of sexual reproduction. So, oogamous type of reproduction. In oogamous type of reproduction, male and female sexual organs are separated. Male sexual organs are called antheridium and female sexual organs are called oogonium or ascogonium. A majority of Penicillium species are homothallic and a few members are heterothallic. By the homothallic species, male and female sexual organs are developed on the same mycelium with different regions. So, that is homothallic species. And heterothallic species. In heterothallic species, male and female sexual organs are developed on two different types of mycelium. So, that is called as heterothallic species. In Penicillium, a majority of species are homothallic. In the vegetative body of mycelium, one of the vegetative cell which is act as a ascogonium. Ascogonium is nothing but it is a female sexual organ in Penicillium. In the developmental stage of ascogonium, ascogonial cell which is mitotically divide and produce two cells, and these two cells repeated mitotic divisions and forms 32 to 64 nuclei. So, this is ascogonium development in Penicillium. Now, the next antheridium. In Penicillium, male sexual organs are called as antheridium. By the time of ascogonium development, one of the vegetative cell which is act as a antheridium. So, this antheridium which is flask shaped or club shaped antheridium. So, these antheridium and ascogonium both are present on the same mycelium with different regions. Ascogonium which is present on the tip of the mycelium and the antheridium which is present below the ascogonium. Fertilization. At the time of fertilization, antheridial cell elongates and spirally coiled and reach the ascogonium. Now, the end of the antheridial cell which attached to the ascogonium. During the fertilization, ascogonial cell and antheridial cell both are fused. At that time, a few enzymes are secreted. With the help of these enzymes, dissolves the septal wall. Now, in this time, antheridial nuclei migrates into the ascogonium and the protoplasm also migrates into the ascogonium. So, in this stage, fusion of protoplasm, that is called as plasmogamy. Plasmogamy which is takes place. Then next plasmogamy which is followed by karyogamy. It means after the fusion of protoplasm, Antheridial nucleus fuses with ascogonial nuclei. Now in this stage a dikaryotic stage which is appeared. Now this dikaryotic stage of mycelium undergoes to 2 meiotic divisions and 1 mitotic division, and finally they forms 8 haploid nuclei, and these haploid nuclei with a few amount of protoplasm which are fused and forms a spore like structures. Now these 8 ascospores are fused in a specialized structure that is called as clistothecium. In clistothecium stage 8 ascospores are present inside the clistothecium. In this clistothecium, when the maturation of ascospores, now the dissolve of peridial wall and these ascospores are liberated, and these ascospores carried out by wind and fall on the suitable substratum, they can germinate and produce new mycelium. So this is sexual reproduction which takes place in Penicillium. So this is life cycle of Penicillium. In this life cycle we are discussed only asexual and sexual stages. In asexual stage, first the vegetative mycelium conidia are formed on the conidiophore. So these conidiophores are fall on the suitable substratum, they can germinate and produce new mycelium. So that is asexual life cycle. During the time of favorable conditions are appeared, at that time Penicillium reproduces with the type of oogamous type. In the oogamous type, Antheridium and Archegonium both are sexual organs in Penicillium. So these Antheridium is a male sex organ and Ascogonium are also called oogonium or Archegonium. It is female sex organ. Antheridium elongates and fuses with Ascogonium. At that time both septal walls are dissolves, nucleus migrates into Ascogonium. Now the formation of dikaryotic mycelium. So these dikaryotic mycelium undergoes to mitotic and meiotic divisions and forms a 8 haploid spores containing specialized structure that is called as clistothecium. And this clistothecium which is covered by a special type of layer that is called peridium. So peridium which is protect the 8 ascospores inside the clistothecium. When the maturation stage peridial wall dissolves, these ascospores fall on the suitable substratum, they can germinate. At the time of germination, a germ tube which is formed inside the ascogonium. Now this germ tube elongation and they repeatedly branched and forms vegetative hyphae. Further development of vegetative hyphae, aerial hyphae, conidiophores and sterigmata and conidia are formed after the development of aerial hyphae. So this is life cycle of Penicillium. And next we are discussed what is use of Penicillium. So these Penicillium species are mostly 146 species are reported. In that, a majority of species are harmful, but a few species are very useful. In that, Penicillium camembert and Penicillium roquefort, these are used for the production of cheese, Swiss cheese and camembert cheese, and also called as blue cheese. And a majority of organic acids such as citric acid, aspartic acid and tartaric acid. So these organic acids are produced with the help of penicillium, and a few enzymes such as lipase, protease, amylase, these enzymes are produced from the Penicillium species. These enzymes used in the formulation of detergents. Next, a few members of Penicillium are used in pharma industries for the preparation of cardiovascular drugs. Penicillin, it is a antibiotic which is extracted from the species of Penicillium chrysogenum. Earlierly, so these chrysogenum which is called as Penicillium notatum. So these penicillin is a very important antibiotic.